Uh, what Memverge was at BioIT World a few weeks ago, and we had a number of really great conversations with end users, and we wanted to, we wanted to bring someone on from the perspective of some of our end users to, to talk about how technologies like, what Memverge is developing, how it integrates and is used in everyday work. And so with that, I wanted to pass it on to Zhong for his introduction and to talk a little bit about metagenomics.
Uh, Hi, uh, thank you, Jing. I'm Zhong. Where I work is not very important here. I have been working in the bioinformatics field for the past 20 plus years, and with 10 plus years in metagenomics, and also experience with AWS. So I'm going to talk about three things today. So first, I'm going to share a little bit what I'm doing with Metagenome, and will also substantialize what the grand challenges in life science Google just talked about from a metagenomics perspective. And then I will daydream an ideal solution. I don't have any solution to offer, but that's what I hope MemWord and its partners can achieve. So I will start with metagenome. So a lot of you, if you haven't heard about metagenome, you should. It's really not really everywhere now. We know, a beautiful looking person can actually also compose of many, many different microbes coexist with this person. And their number is actually 10 times of the number of cells a human has. So you are pretty safe here in the Zoom setting because as I speak, I'm also exchanging microbes with my environment. So you're not in my room, so you're safe. You're protected from the microbes I'm spreading. So the microbes, they really play an essential role in many human aspects, from the medical, nutrition, and also sometimes psychological too. So I'm not going to any details of those. I would rather just focus on some general aspect of metagenomics. So normally, as a scientist, we ask three types of questions in the metagenomics, which is the study of microbial communities. So we want to know what kinds of microbes are there, and we want to understand what they are doing. So these two, the first one, solve the diversity problem. We know there are a lot of novel microbes out there that are yet to be discovered. And the second question, we are really asking what kind of metabolic capabilities they uniquely have that we can leverage to improve our own life. And finally, we want to understand how they form a stable community by interacting with the environment or with each other. Next slide. So I was going to share two research stories I have been working. So the first one, we want to improve the biofuel research. So one of the key steps in biofuel research is the high efficient cellulases, which convert biomass into energy. So we can, like ethanol, we can pump into our car. So to study this, we look around our environment and we ask, where we could find high efficient cellulose. We know that cow, even they don't encode any cellulose gene in their own genome, but they can harness the microbial power in their rumen, the largest stomach in the gut, to degrade, to help them to digest the grass they eat. So in this experiment, so our technician stick their hands inside the cow and pull out the microbes that work on the cellulose fiber. And then we did a metagenomic study. And in this single study done more than 10 years ago now, we identified thousands of novel cellulose candidates. And the number actually compared to all the known cellulose people have found in the past 30 years. And next slide. So in this experiment, we didn't pull microbes out from the cow. We actually pull microbes out from 8 meters underneath the ice, thick ice in Antarctica. So our collaborator went to Antarctica, drill a hole, and to isolate microbes that have been isolated from the outside world for hundreds of years. So in this, we're hoping this place can give us many novel microbial species. So and turned out after we got the data, 99% of the species we found underneath the earth are novel, and we're only able to put 40% of these microbes on the phylogenetic tree, and we're trying to try to annotate, and we were only able to annotate about 80%, less than 80% of the microbes, and there are quite a few microbes that we have never seen before, and we have no idea what they're doing there. So next slide. So I made the metagenomic study sounds very easy, but in order to do the above two studies, we'll have to take several steps, this including experimental steps and also informatics steps. So experimentally, we have to isolate the microbes from the environment, and then we'll extract their DNA. And because we don't have a sequencing technology that can read the genome from beginning to the end, we break the genomic DNA into short pieces and end up with a pile of short reads, mixing all the different species. And then we use a process called metagenome assembly that we can predict or reconstruct the microbial genomes from a mixture of metagenomic data. So in the past 10 years, I have been working to improve the metagenome assembly process. Next slide. So, so from a computational metagenomic perspective, we're actually facing many challenges. Google touched base of this big data, and our data is also very noisy. We have contamination, we have biases, also some of the microbes are missing from the data. So we constantly deal with this kind of problem. There's also, as in other fields, in bioinformatics, many of our software algorithms are not reliable. They actually, by using them, they also introduce bias and noise. So sometimes we have to combine different algorithms and try to solve the same task and protocol ensemble. So these two aspect of challenges, they both increase the running time of our workflows, and also different algorithm also introduces the different various resource demanding steps in the workflow. So it sounds like the data science can do all these cool things, find novel enzyme, find novel species, but in reality, when you first started with computational metagenomics, you find that you're more like playing a one-man band. You have to think about, how to import and export the data, how to store the data safely, how to find the reliable algorithms, how to satisfy the various demanding apps in your workflow. So it's not very fun. Yeah, next slide. So I just use one example, the metagenome assembly workflow. We do this routinely. So in this complex graph, just pay attention to the color. So the different background color are different apps in our workflow. And there are two important lines. So there's lighter blue line, that's the CPU usage during the workflow, and the dark green line, that's the memory usage of the workflow. And you can see, and different steps can have dramatic different CPU and memory requirement. And not only that, there's a single step like the light green background step, that's actually the metagenome assembly step, more specifically metaspade, that the app itself during different stages, it has very different CPU and memory requirement. And current strategy, we're trying to break different apps using different containers and provision with the flexibility of AWS provide into different size of containers, right? So you can optimize the workflow a little bit. But since, like the metaspace step, it itself has to be in one container. It has various steps. So it next. So of course, everybody's familiar with out-of-memory error, right? So a lot of our bioinformatics applications require a lot of RAM. And these also make things a little bit worse because some of the data sets are just inherently more complicated than others. It's very hard to predict before you run the workflow how much RAM you will need. A lot of people just use as much memory as big an instance they could find to run the workflow. Next. Yes, the Spot instance Google just mentioned of AWS can save us a lot of computing cost. But in reality, when because everybody trying to use the Spot instance, and then sometimes the limit size of the pool of the Spot instance can actually add challenges to long-run jobs. So this is just one example. I'm not going to talk details about what workflow this is, but in this graph, we just plot the node addition and the node removal. So node addition is light blue, and node removal due to Spot reclamation in red. Yeah, you can see, we have a lot of, we lost a lot of Spot instance in the duration of this workflow. Actually, this actually is a little bit counter productive now. We had to use like on-demand instance for this job. Next. So now I start to daydreaming. So to solve the one-man band problem in data science, and especially in metagenomic field, we would like a product that is, first easy to use, because we want to spend most of the time developing new algorithm, not to just fight, the user interface, the hardware software configuration. And we would like to, as a scientist, we want to reduce our research cost, and we like to know even before we run the workflow how much this will cost. And of course, we will use as much as we could to reduce the total cost. And finally, if we have to use Spot instance, we would like have a robust solution, especially as many of our workflows will run a long time. So the ideal product should safeguard these unpredicted events like Spot reclamation. So that's all what I have. If you have any questions, feel free to put them in the chat.